Good afternoon. Last time we talked about getting nucleic acids out of the virus particle, destabilizing it. So when we talk about metastable, we actually covered metastability in 2 lectures. We talked about stability when we talked about virus structure. We talk about the instability when we talk about uncoding. So that should make it clear to you. And now we're going to start moving through the replication cycle or the reproductive cycle. We've got our nucleic acid in the cell, and we're going to talk about what happens all the way through the end of the cycle. That'll bring us to the first half of this course. And today we're going to talk about RNA viruses and how they make mRNAs and more genomes. So this is called RNA-directed RNA synthesis. A little bit of RNA history just for your enjoyment. You don't have to remember any of this. You can sit back and just listen and look. 1935, remember, tobacco mosaic virus figures in lots of landmarks in the history of virology. It was the first virus ever discovered, right? In 1935, Stanley crystallized this virus. He made crystals of tobacco mosaic virus, and a year later found that they have about 5% RNA in them. So this gets people thinking, what is this RNA doing there? And this is 1935. 1936, we know what nucleic acids are, but we don't yet understand what they're doing. 1944, the famous experiment showing that DNA is genetic material. Followed in 1952 by the Hershey-Chase experiment showing that DNA is the genetic material of a bacteriophage. Structure of DNA, 1953. It wasn't until 1956 that it was shown that the RNA of tobacco mosaic virus is infectious. So Frankel Conrad purified the RNA away from the protein. He put each into cells and showed it was the RNA that initiated an infectious cycle. First demonstration that RNA could be genetic material. And that opened the floodgates. By 1959, many viruses were shown to have RNA, and people were interested in how does this RNA get copied? And at this point, no one knows anything about viral RNA-dependent RNA polymerases. So all they know is what's in cells, DNA polymerases that copy DNA, transcriptases that make mRNA from DNA. That's all we know. We don't know. So people are very interested in understanding how RNA of RNA viruses would get copied. One of the ideas was that it was copied by a cellular DNA-dependent RNA polymerase in the absence of any other evidence. So today we're going to talk about RNA synthesis of viruses, and going back to our Baltimore scheme, we're going to talk about the viruses in red today. So we're going to talk about plus-strand RNA viruses, and I'll mention polio quite often as our type virus. These are viruses with plus-strand RNA genomes. We're going to talk about 2 negative-strand viruses, influenza virus and vesicular stomatitis virus. You should be hearing the word influenza virus a lot these days, right? There's a serious outbreak going on this season. And we'll get to talking about disease later on. We're going to also talk about Reovirus and rotaviruses, viruses with double-stranded RNA genomes. We're going to look at how these viruses make mRNAs and how they copy their genomes. Getting back to 1959, a graduate student here at Rockefeller University, David Baltimore, that name may ring a bell. He's the one who made the Baltimore scheme, but he started out trying to understand how RNA viruses replicated their genome. And as I said, at the time, people thought it was a cellular polymerase. So he did a very simple experiment where he took poliovirus, he infected cells, and then after different times after infection, he would make extracts from the cells. He would add triphosphates, one of which was labeled with a radioactive isotope. And then he would incubate this mixture and ask, can I find RNA being synthesized in this extract? The reason he broke the cells open is because you can't add triphosphates to cells. They won't get through the membrane. Later on, we figured out how to add precursors to cells, but now he had to break the cells open, make an extract, and measure RNA synthesis. And this is his graph. You can see it's one of those old hand-drawn graphs. In the old days, before computers, we had to draw our own graphs. This is from an old paper. Anyway, what we're seeing here, we're looking at hours post-infection, so 0 to 6, a very short infection, typical of poliovirus. And what he did for each time point, not only did he measure the incorporation of label into RNA, but he measured poliovirus production, PFU per mil, dotted line. So you can see the lovely eclipse period. This is total virus production in the, he would just break the cells open and measure virus production. Nice little eclipse period where the genomes are being expressed and proteins are being made. And then at about 2 hours, you see, between 2 and 3 hours, you see the first virus made. Then you have a peak at 5 or 6 hours post-infection. At the same time, he measured RNA synthesis, and that's the solid line, and that's shown on the other y-axis. RNA polymerase activity counts per milligram of protein, which was a standard way of doing this back then. And you can see that line parallels that, the solid line parallels that of production of infectious viruses. So there was RNA synthesis going on in these cells. Of course, this doesn't tell you if it's viral or cellular, right? It doesn't tell you what kind of enzyme is doing it. But he did another experiment in this same paper where he added a drug to cells which inhibits DNA-dependent RNA synthesis, a drug called actinomycin D. And it had no effect on the production of this RNA in infected cells. Now, this is viral RNA, we know, because it's not on in the early times after infection. There's no incorporation. So it's not just the cell making RNA from something, it's the virus. And he showed that it was resistant to a drug that inhibits the cellular DNA-dependent RNA polymerase. So that said, it wasn't a cell enzyme, it must be a viral enzyme. And he continued to work on that for many years. He worked on poliovirus polymerase, he moved to VSV, and showed that the polymerase was in the particle, and eventually moved to retroviruses, discovered reverse transcriptase in the particle, got the Nobel Prize for that. So from very humble beginnings like this, he went pretty far. As I said, David Baltimore said, this is an RNA polymerase, and then he said, you know, these viruses with negative-strand genomes, I'll bet there's a polymerase in the particle, because if that RNA gets into a cell, it can't be translated or replicated because there's no cellular polymerase. So he found the polymerase in the particles of negative-strand viruses. So here's vesicular stomatitis virus, our bullet-shaped negative-strand RNA virus. He purified these virus particles, cracked them open slightly, and added the labeled triphosphates and found that there was a polymerase in the virus particles. From that came our idea that plus-strand RNA viruses, there's no polymerase in the particle because the RNA can be translated. As soon as it gets in a cell, negative-strand viruses need to have a polymerase in the particle. Years later, people began to sequence viral genomes and look at the genes encoding these polymerases, and then you could simply look at the amino acid sequence of an RNA-dependent RNA polymerase and say, this is a polymerase. Why? Because there are certain motifs like gly, asp, asp, 3 amino acids, GDD, which are diagnostic for being a polymerase. And then people could take the gene encoding these proteins, express it in cells, and show that you could make a polymerase apart from all the other viral proteins. And then, of course, eventually, people wanted to know the 3-dimensional structures of these polymerases. Just like people wanted to know the 3-dimensional structures of virus particles, which we went through a bit. People wanted to know polymerase structures, and they solved them by X-ray crystallography. And I'll show you some of those today. So this brings us to a kind of a summary of the state of the RNA in the virus particle of these various viruses. So we have viruses with negative-strand RNA genomes. The RNA in the particle is typically coated with protein, and we show that for VSV and influenza virus. And that's where this helical nature of the nucleocapsid comes into play. These RNAs are not naked, they're coated with protein, and the protein wraps them out around them in a helical fashion. And this is related to the need to make RNA, to bring in a polymerase and make RNA as the first step in infecting. And in these viruses, there are also polymerase, RNA-dependent RNA polymerases in the particle, and you can see that there for VSV, that yellow molecule. And there's one attached to each of the 8 RNA ribonucleoproteins of influenza virus. Plus-strand RNA genomes are mostly naked. There are a few exceptions. Poliovirus, for example, Flaviviruses, and many others. There's just naked RNA in the capsid, and there's no enzymes, there's no proteins coding the genome. That RNA comes into the cell and it's immediately translated. So there's no need for there to be an RNA polymerase in the particle. Of course, there are always exceptions. The exceptions for these plus-strand viruses are retroviruses. These are plus-strand RNA viruses with a DNA intermediate. We're going to consider them in their own session. Those plus-strand genomes are not translated when they enter cells. Instead, they're copied into DNA. And they're also coded. Here's a retrovirus particle, third from left. They're also coded with proteins in the virion. And the coronaviruses on the right are also plus-strand RNA viruses, but their genome is coded with protein. They don't bring in a polymerase, but their genome is coded with protein. When that genome gets in the cell, the first step is it is translated. And we're not sure exactly why the genome is coded with protein. It turns out that these viruses have the biggest RNA genomes of any of the viruses. They're well over 20, 25, 30 kilobases, maybe even more in length. And maybe this is in part to protect the RNA from breaking. And now, at the bottom are the viruses with double-stranded RNA genomes. These double-stranded RNAs are actually naked in the particle. They're not coated with proteins. But remember, I told you a few sessions ago that a double-stranded RNA, even though it has a plus and a minus strand, the plus strand can't be accessed by ribosomes. So there has to be an RNA polymerase in the particle of these double-stranded RNA viruses so that those double-stranded RNAs can be transcribed into mRNA when they come in the cell. That polymerase is not coding the double-stranded RNA. Rather, there's a molecule of RNA polymerase at each 5-fold axis of symmetry. So remember, these are double-shelled particles. At each 5-fold axis, you see there's a kind of a yellowish-orange structure here called a turret. You'll see later today why that's called a turret. But right below that turret, there's a molecule of RNA polymerase at each 5-fold axis of symmetry. Now, just to review the negative-strand RNA structures, remember on the left, VSV, an enveloped negative-strand virus with a helical nucleocapsid, one protein repeated many, many times, which binds its neighbors and the RNA at the same time. And for VSV, it's called the nucleocapsid protein. We were first introduced to this via tobacco mosaic virus, a similar coat protein binding the RNA. But for VSV, it is an enveloped virus. And here on the top shows you these are 3 nucleocapsid monomers binding a piece of RNA. And that's the actual X-ray structure. And the point is to show you how these proteins interact with each other as well as with the RNA. So there are places on these proteins for protein-protein interactions as well as protein-RNA interactions. And of course, these interactions make this stable. That's what gives the stability, this symmetrical repetition of proteins. And the result is this lovely helical bullet, which happens to be closed at one end. On the right is Influenza virus, another negative-strand virus with a helical nucleocapsid. In this case, it's segmented. There are 8 pieces of RNA, each encoding 1 or more proteins. And those RNAs are bound up with protein, just like the VSV nucleocapsid is. On the top is the structure of one influenza virus RNA bound to these nucleoproteins. And again, both helical, although it may look more obvious in the case of VSV. Now, RNA is not just linear strands. As I told you earlier, it has lots of secondary structure. And this is important for the function of the RNA. When it's replicated, often polymerases and other proteins recognize these structures. So I just want to mention them again because we'll refer to them throughout the first half of this course. RNA, shown here, plus-stranded RNA, can fold into secondary and tertiary structures. So the primary structure of an RNA is its sequence, of course. The secondary structure is how it can fold by making these various stem loop structures. You can see a stem here, which is formed by base pairing when sequences in the RNA are complementary to one another. The red bars indicate that there is base pairing, so there's AU base pairs, CG base pairs. And there can be complicated types of stem loops. You can have extended structures, as you see here, with multi-branched loops, interior loops, bulges, hairpins, and so forth. And these often are the binding sites for proteins. But RNA can also interact with other RNA sequences, either short or long distance. And we talked about a long distance interaction in a plant virus RNA where loop regions can interact from the front to the end of an RNA molecule. Here's a more short distance interaction. Here is a stem loop structure shown in B, where bases in the loop are actually base pairing with sequences just downstream of the stem loop. All right, so the dotted lines show you the base pairing that's happening. What that does is make an interesting structure. So in the next panel, you can see how that actually looks like when both the stem and that loop region are base paired. So the loop is kind of folded down onto the RNA. And then at the bottom here is the actual tracing of the backbone of the RNA. It kind of twists around itself. On the upper right is the actual three-dimensional structure of this. And it's called a pseudoknot, because it looks like a knot. It isn't actually a knot like you would tie a piece of string into a knot, but it's got this twist back structure here called a pseudoknot. You'll see us talk about that in various replication schemes. And this is often an important point for the interaction of proteins with the RNA. So we have some rules for viral RNA synthesis that are important. There's a typical single-stranded RNA. It has a 5' and a 3' end, of course. First, the RNA must be copied end to end. You can't lose any sequence. You can't start a little bit from the end and end a little bit from the other, and that wouldn't work. Eventually, you'd lose sequence and you wouldn't be able to replicate anymore. So we have to go end to faithfully reproduce the genome. And secondly, viral RNAs have to be made that can be translated by the host cell ribosome. You can't make an mRNA that won't be recognized by ribosomes. All right, so end-to-end replication and a good mRNA that can be recognized. Now, all RNA-directed RNA synthesis has universal rules. Some of them resemble those for DNA that we'll talk about in a couple of sessions. First, the RNA synthesis begins and ends at specific sites on the template. It doesn't begin just anywhere, and it doesn't end just anywhere. There's specific sites. The initiation of RNA synthesis can occur de novo, and this is how DNA-dependent RNA polymerases work. The enzyme that makes mRNA in your cells does not need a primer. That's what de novo means. But some RNA-dependent RNA polymerases do require a primer. So you have both. They can either initiate de novo or they may need a primer, and we'll see some examples of that today. It's not just the polymerase molecule that will work to copy RNA. The RNA-dependent RNA polymerase often requires other proteins. These can be both viral and cellular proteins, and it's very rare that you could take just the purified polymerase, RNA-dependent RNA polymerase, and get enzymatic activity from that. The way you make RNA, of course, is it's template-directed. Okay, it's got a template that it's copying. It's not making RNA on its own, although there is an example of that we'll talk about. You make template-directed stepwise incorporation of the triphosphates, elongated in a 5' to 3' direction. So you copy the template from 3' to 5', you make a new strand in the 5' to 3' direction. There is some non-templated RNA synthesis. I'm not sure we'll talk about it in this course, but it is basically when a polymerase is copying a template, it stops, and it starts to churn out bases that are not templated. Actually, we will talk about it today, in fact. And that's called non-templated because it's not copying anything in order to make that. All right, so those are the universal rules for RNA synthesis. Let's look a little bit at initiation steps so that you understand conceptually what's going on here. We have the two kinds of initiation: de novo, where we don't need a primer, or primer dependent on the bottom. So de novo, basically, the 3' end, which is where copying would start, the polymerase puts down that first NTP complementary to the last base at the 3' end of the RNA. Very simple. Polymerase recognizes the end, starts at that very 3' base, and begins to copy it. You can also have an internal initiation. In this example, the polymerase has started polymerizing at the 6th base in from the 3' end. You can see that GC pair begins to elongate. However, if you didn't copy the very end of the molecule, you'd have a problem eventually. As I said, you have to copy end to end. So in these cases, the polymerase actually slips back and copies the rest of it. And the sequence is constructed so it could slip back, that this product can actually slip back, and it will be complementary and continue to polymerize. So the point here is that even if you don't start at the very 3' end, you'll eventually have to slip back and fill it in. At the bottom, primer-dependent initiations. We have two examples. Some viruses require a protein linked to a triphosphate as a primer. That's called the terminal protein. And some viruses require a capped primer. As you know, all mRNAs in our cells have a cap at their 5' end. It's a very special way of attaching a base, and we'll look at that later on. Some viruses actually require a capped primer, as a capped piece of RNA as a primer. So how does polymerization work? I'm sure you all learned this at some point and forgot it afterwards, after the exam. And this is the two-metal mechanism of polymerase catalysis, devised many years ago for DNA polymerases. It also works for RNA polymerases. Here the figure is drawn from DNA, which is why you'll see T's instead of U's, but the mechanism is the same. Again, we have a template shown at the upper left in the 3' to 5' direction. The polymerase, shown as a DNA polymerase here, but could be an RNA polymerase, is in purple color. It's copying the template and synthesizing a copy of it in a 5' to 3' direction. And this particular enzyme requires a primer, which is shown in red, and then the polymerase will be adding bases onto the 3' end of the primer so that the strand will grow in a 5' to 3' direction. The way the bases are added is by this two-metal mechanism of catalysis. And I'm showing you this because in a moment I'm going to show you structures of these enzymes and show you exactly where this is. Very neat. So here on the right we have the chemical structure of a template, just a few bases here, 4 bases, 3' to 5'. We have a G, a T, an A, and a T. And you can see that 2 bases have already been added. A C has been added to the G, and A has been added to the T. And of course, these now have a phosphate in between them. It's a phosphodiester bond right there. So there's your ribose sugar, a base is colored, and the phosphate bond. And then we have our next base, of course. And here we have a polymerase in the process of adding a 3rd base to this product. It's adding a T, which is complementary to that A. And you can see that this T comes as TTP, thymidine triphosphate, 1, 2, 3 phosphates attached to it. The polymerase in its active site has 2 aspartate residues, aspartate A and aspartate C. Their function, right in the catalytic site, their function is to coordinate 2 magnesium ions, 2 metals. That's where the name comes from here, magnesium 1 and 2. It holds them via those aspartates, and those catalyze the addition, the chemical reactions that lead to the removal of a diphosphate and the formation of a phosphodiester bond. So you see a series of nucleophilic attacks here, which basically involve that oxygen attacking the phosphorus, the first phosphate attached to the sugar. That's going to form the phosphodiester bond. And in the process, the other 2 phosphates are released, and of course that releases energy because this is a triphosphate, and the energy is used for the polymerization reaction. And this goes on and on, happens very quickly. Very, very many bases per second get polymerized. So both RNA and DNA polymerases do that in pretty much the same mechanism. We have a first question here, which is a universal rule about RNA-directed RNA synthesis? The RDRP may initiate de novo or require a primer. RNA synthesis initiates randomly on the RNA template. RNA is synthesized in a 3' to 5' direction. RNA synthesis is always template-directed. All right, good enough. How do we do? 90% got the right answer. It may initiate de novo or require a primer. Everything else is wrong. It doesn't initiate randomly, which I think you remember I said, synthesized in a 3 to 5 prime direction. No, it's synthesized in a 5 to 3 prime, but it reads the template in a 3 to 5 prime direction. And it is always template directed. Not always. There can be non-templated RNA, which was on this one. See, some non-templated synthesis. Now, when people began to determine the sequence of these RNA-dependent RNA polymerases, and the other polymerases as well, there are 4 classes of nucleic acid polymerase. There is what we're talking about today, RNA-dependent RNA polymerase, which is exclusively viral. There's RNA-dependent DNA polymerase. This is reverse transcriptase, which is not exclusively viral. There's DNA-dependent DNA polymerase, which copies our DNA and that of viruses as well. And there's DNA-dependent RNA polymerases, which makes mRNA and other kinds of RNAs in cells. And when people sequenced representative of each one, they realized they're all related. They have sequence relationships. They have some homology. So these are just bar diagrams indicating the amino acid sequence of the 4 kinds of polymerase. And these colored areas are areas of significant or substantial identity among the proteins. So you can see they have very similar organizations, and in a moment you'll see they all look very similar. And they probably evolved from a common ancestor. There was probably one nucleic acid polymerase many years ago, and that diversified in the kinds of templates it could copy. Now what we see here are some interesting areas. They're colored A, B, C, D, and E. And in a moment, I'm going to show you the 3-dimensional structure, and you'll see these in the context of 3 dimensions. And those, again, are the conserved regions among these, the most conserved among these polymerases. The one that I want to point out here is motif C. And this is where there can be a 2 or 3 amino acid sequence that will tell you, if you see it, that this is probably a nucleic acid polymerase. So the GlyAsp that I mentioned, GDD, that's found in there, and the asp in the reverse transcriptase. Actually, these are the 2 asps that coordinate the magnesium during the 2-metal catalysis of polymerization. That's why they're conserved, because all these polymerases do the same thing. In non-segmented -strand polymerases, happens to be a GlyAsp. So you can look at these signatures and say whether or not it's polymerase from seeing them. So here's a 3-dimensional structure of the RNA-dependent RNA polymerase of poliovirus, solved a number of years ago. And all these polymerases, by the way, resemble a hand, in particular a right hand with fingers, palm, and thumb domains. And I've colored them on this little diagram of a hand to match these structures. So the blue parts are the fingers, and the yellow, the palm part, is the active site, shown right there. It's a bit of a stretch, frankly, to call it a hand, but crystallographers are weird, so that was their doing. But if you think of it as that, the palm is the active site. That's where the nucleic acid is going to be polymerized. So here we have the fingers and thumb domain of the polio polymerase. And these motifs are colored, like motif A is in red, and so forth. Green is here, motif B. But the one I want to show you is motif C, which is making up the active site right there in the middle of the molecule. And I've inserted the side change of the 2 aspartates. You can see them there. And those are the conserved part of the GlyAsp, and those are the 2 amino acids that hold on to the magnesium ion during polymerization. So they're really important. So right there is where the activity of this enzyme is, and it's in the palm domain. And here's a space-filling view of the polio polymerase. I've actually removed some amino acids here in the front so you can see what's going on. I have now colored those 2 aspartate residues in cyan, and you can see one magnesium ion right there, the little red sphere. The other one is hidden. So that's the active site where the aspartates are holding the magnesium to assist in the polymerization. And what we have here is we have a template in cyan. That's cyan. Sorry, the aspartates are magenta, right? It's not cyan. And the template is shown in cyan, and it goes in the front of the molecule here. You can see it's snaking through the active site, and it will come out the other side. And as this snakes through, the polymerase is making a product, which is shown in yellow. So it's using the template, of course, to read the sequence. It's making a product, and then out comes both the product and the template. So the RNA snakes in and out of the polymerase. And of course, all this is covered. You would not be able to see this. Normally, again, as I say, we've removed some amino acids to show you this. So it's a really amazing molecule, and all of this happens very quickly to polymerize these RNAs. Now, a very interesting finding came out of this structure, or many interesting findings, but here's one I want to share with you. It's why the RNA polymerases use NTPs. They don't use dNTPs. That's what makes them an RNA polymerase. So here is a close-up of the active site. Here are 2 aspartate residues, the ones that coordinate the magnesium ions. Here is a UTP. So the structure in this case was determined with a UTP, a uridine triphosphate bound in the active site. So you can make crystals of your protein and you can soak them with UTP, and the UTP will go to the active site. And you can see it's being held in place by hydrogen bonding with amino acids of the polymerase. They're shown here, labeled, and also as part of the red motif. And in particular, this one here, this hydrogen bond here between the 2' hydroxyl of the sugar, the ribose of the UTP, that's the one on the left, and aspartate at 238. It's a different one from the yellow aspartate. That hydrogen bond would not happen if this were a dNTP. There wouldn't be an oxygen on the ribose ring. It would be a hydrogen. It wouldn't form a hydrogen bond with that aspartate 238, and it wouldn't be incorporated. So that simple interaction determines whether this is an RNA or a DNA polymerase. So I think you can see that it only takes one amino acid, or maybe just a few changes, to take one kind of enzyme that recognizes and makes DNA to another kind that would make just RNA. It's a very simple interaction there. So how does this work in the big picture? Let's step back and look at the scheme. We're going to look at 2 kinds of plus-strand RNA viruses because they illustrate different principles. On the left are the plus-strand RNA viruses, which would include Poliovirus, the picornaviruses, that's the family name for Poliovirus, rhinovirus, and related viruses, and then the Flaviviruses. They're both plus-strand RNA viruses. The Flavies, of course, dengue virus, Zika virus, West Nile virus. And those are shown at the bottom. Polio on the left, Flaviviruses on the right. They have plus-strand RNAs. The Flavi RNAs are capped, but the polio RNAs are not. So this is not correct for polio. It would be a protein there, actually, at the 5' end. But the idea is correct. The 3' end is polyadenylated. These RNAs come into the cell. They're translated to make an RNA polymerase that replicates the genome, and replication makes more genomes as well as mRNAs. For these viruses, the genome is the same as the mRNA. And you can see for the Flavies, it's even capped. So how does that work in an infected cell? Here we'll look at poliovirus. The virus binds receptors, taken into the cell. We saw last time how the RNA gets out of a pore in the particle very early. It doesn't need to go through the whole endocytic pathway. No acidification required. Now we have the RNA in the cytoplasm. Step 3, it's translated immediately. Cellular ribosomes bind to it, make proteins. Those proteins go on to replicate the viral RNA. So among the proteins are the polymerase, and that's shown here on the right-hand side. The RNA synthesis happens on membranous vessels that are actually induced by virus infection. Many RNA viruses do this, and it may be to shield them or make the reactions more efficient. But you can see the plus strand going to plus minus, and then minus to more plus. And eventually you'll have proteins made to assemble new capsids, and these would exit the cell. So very straightforward replication. The polio genome, just so that you know, here's the plus-strand RNA at the top. It has a protein on its 5' end, not a cap as was shown in the previous picture. It has a long RNA, about 7,500 bases, and then there is a poly-A sequence at the 3' end. And the way this is expressed is interesting. It's made into one long protein. Ribosomes bind near the 5' end, and they make one long protein. And then within that protein, there are embedded proteases that will chop it up into individual proteins that you see at the bottom here. So very interesting strategy. In eukaryotic cells, it's hard to make more than one protein from a messenger RNA. And this is one way to get around it. You make a precursor that can be cleaved and chopped up by proteases, and many viruses do that. And so the proteases are embedded in the viral genome. In fact, for polio, there are two of them, 2A and 3C. It's not important for you to know what they are, except that they're proteases that will process this and give you the proteins you need, including the capsid proteins, the VPs, and down here, 3-dpol is the name of the RNA-dependent RNA polymerase. So that's how the proteins are made. Now let's look a little bit in detail at the mechanism of poliovirus RNA synthesis. Here's the very 5' end of the viral RNA. It's the first 120 bases or so. And what we find here is our protein. This is a viral protein, 23 amino acid protein called VPG, which is covalently linked to the first base in the viral RNA. It happens to be a uridine. And then the rest of the genome, the rest of the 120 or so bases, you can see is folded into this cloverleaf structure. So there are 4 stem loop structures. And this turns out to be very important for the replication of this genome. In the cell, this protein is actually removed. So as soon as the RNA gets into a cell and is released in the cytoplasm, this protein is removed by a cellular enzyme. The reason it's there is because it's a primer for RNA synthesis, but you don't need it once the RNA gets into the cell. So here's an overview of the entire viral RNA, now showing secondary structures that we know are important for replication. So we have at the 5' end, that protein linked to the RNA. We have this cloverleaf structure. Then in the middle of the genome, we have another stem loop called the CRE. CRE stands for cis-acting RNA element. That means it's operative on the same genome as something else that's interacting with it. You'll see what that is in a moment. This can be pretty much anywhere in the genome because it's going to act as an interactant site for the polymerase. And then at the 3' end, we have a pseudoknot and then the poly A tail. Now these are all important signals for directing the RNA polymerase of the virus to this RNA. And these signals are not present in cellular mRNAs, and that's why they're not copied by the polymerase. If you infect a cell with poliovirus, after a few hours, you're churning out lots of viral RNA, as we saw in one of the first slides. No cellular RNA is ever copied by the polymerase. Exquisite specificity, which is good. It makes sense because it's efficient. You're not going to copy cell mRNAs. That would be a waste of resources because they're not going to be packaged, and even if they were, they wouldn't help the viral infection. So these signals, cloverleaf, CRE, pseudoknot at the 3' end, they all give specificity to the polymerase recognizing this RNA. Now, what's the role of VPG in this process? It involves CRE. So now we're just looking at the CRE element. I've removed it from the genome. And what we think happens is that the polymerase is produced as a precursor with the protease. So the polymerase, the RNA polymerase is in blue, the protease is in red, and that binds the CRE element. So that's why we call it a cis-acting element. This polymerase is produced from the genome and then binds to a CRE element on the same RNA. And then a polymerase molecule binds to the loop at the top. 3D is the other name for the RNA polymerase. VPG comes in, so VPG has been synthesized in the infected cell. That binds into the polymerase, and then two U's are added to the VPG molecule. They're covalently attached, and they're templated by a series of A residues in this loop of the CRE structure. So there's a series of A's up here, and that's the template the polymerase uses for the addition of UU to VPG. And this will be the primer for RNA synthesis. So polio polymerase is a primer-dependent enzyme. In fact, all the picorna polymerases are. The primer is VPG, a viral protein linked to two uridine residues. All right, and this is how that works. Here's the initiation step of RNA synthesis. This is all membrane bound. So here's a membrane at the upper left. There's a viral protein called 3AB embedded in the membrane, and that attracts the viral RNA. So the viral RNA is shown with its cloverleaf, CRE element, pseudoknot, and it's also shown binding a protein called PCBP. This is a cellular protein. poly-C binding protein. It was discovered because it binds poly-Cs, but it also binds this clover leaf, attaches the RNA to the membranes, and in turn, the poly-A tail binds poly-A binding protein, which is another cellular protein. PABP in turn binds the clover leaf, so now you've circularized the RNA in effect, and now we have a polymerase coming in here, 3CD-Pro. VPG-UU binds to A's at the 3' end of the RNA. That's the priming step, and then it elongates the negative strand, which you can see at the bottom here. So a couple of interesting things. First, two cellular proteins are absolutely necessary for this, PCBP and PABP, and they function by circularizing the RNA. The RNA is circularized. It's very interesting. So the polymerase binds to the 5' end, but it initiates at the 3' end because the 3' end is brought around to it. And finally, you make a double-stranded intermediate, which is then copied again to make more plus strands by a similar mechanism. So that's how the primer works in that replication scheme. Now, as I mentioned, this RNA synthesis typically occurs on vesicles that are induced by virus infection. These vesicles are not present in an uninfected cell. So on the upper left is an uninfected HeLa cell, and you can see endoplasmic reticulum. There are mitochondria, Golgi. If you infect with poliovirus, after a couple of hours, the ER, the Golgi, is gone. All those membranes have been repurposed to form these membranous vesicles, the circular guys here, on which the viral RNA synthesis occurs. And this doesn't just happen for picornaviruses. The membranes are induced in flavivirus infected cells. You can see them here. They're derived from the ER, and they are the sites of viral replication. In fact, the virus particles here shown in red actually assemble on these membranes. And a coronavirus infected cell, you can see all the membranes induced there as well. So many plus-strand RNA viruses induce these membranes. We think they're important for allowing efficiency of the RNA polymerization reaction. And they may also shield this RNA from detection by the innate immune system. Okay, membranous vesicles are important for RNA synthesis. Next question is, which is a part of the poliovirus replication strategy? A, the production of subgenomic mRNAs. Subgenomic would be shorter than the genome. B, de novo, without primer, initiation of RNA synthesis. C, circularization of template for initiation of RNA synthesis, or all of the above. So about half of you got C, circularization of template, which is the correct answer. Many of you picked all of the above. However, subgenomic RNAs aren't part of the strategy. I'll go back and look at it and show it to you again so you can see that. And we do have a primer. It's VPG linked to UU, right? So that's the primer for RNA synthesis. So let's go back and take a look at that. So here the primer is VPG UU. And you make a complete copy of the input. So the input is the plus strand on top, and the copy is the negative on bottom. It's a complete copy. It's not a subgenomic RNA. However, that leads us to the next virus, which does make a subgenomic RNA. So that's a viable strategy. So on the left, Picornas flavis, they don't make subgenomic RNAs. The mRNA is the same as the genome. Here we have other viruses, which we've not mentioned in this course, but they are called Alphaviruses or togaviruses. They include chikungunya virus, which you may have heard of, a mosquito-borne virus that causes arthralgias and rashes, very much like Zika. These viruses have plus-strand RNAs. They're capped. They replicate via a negative-strand intermediate, but a subgenomic mRNA is made to access the coding region at the very end of the genome. What does that mean? Well, why do we have to do that? Well, let's take a look. So these viruses come in the cells and they enter by endocytosis. Low pH catalyzed fusion sends the RNA in the cytoplasm. These are naked RNAs. They engage ribosomes. They are translated, but only a part of the plus strand is translated. We'll see exactly how much in a moment. That part that's made is actually the polymerase. Polymerase copies the input to make a negative strand, which then, of course, is copied to make more plus strands, and these eventually will go into virus particles. A fraction of the minus strands are the templates for the making of a subgenomic mRNA, shown in step 5, which is needed to make the structural proteins. The structural proteins are encoded at that part of the genome. Why this is this way, well, that can't be answered. It does work. It's a viable strategy, and obviously it has been selected for evolutionarily. And this is a schematic of what happens. Here's the plus-strand RNA at the top. Comes in the cell naked. It's immediately engaged by ribosomes, and they produce the RNA polymerase and some accessory proteins. But the translation stops right at the end of the polymerase. There's a stop codon in the middle of that mRNA. And so the rest of the proteins that are encoded down here are never made. Now, if I were designing this virus, I wouldn't put a stop codon there. I'd make a polyprotein, just like polio and the flavies. But this is the way it evolved, and it works. So the only way you can access the coding region at the 3' end, this polymerase copies the plus strand, makes a minus strand, and then the same polymerase initiates at this red site, this promoter for subgenomic mRNA synthesis. And now that's a plus-strand mRNA capped and polyadenylated that encodes the structural proteins, the capsid, which forms the nucleocapsid, and the glycoproteins. So it's a different strategy for a plus-strand virus, and I show it to you to illustrate that you can have two ways of accessing these big genomes, make a polyprotein or make a subgenomic mRNA. Let's turn to negative-strand viruses, see how they are different. And we, of course, have viruses with a single negative-strand genome or with segmented genomes. Principles are the same, but we're going to look at a few differences. On the left, we have VSV. And again, because these are negative-stranded, the negative-strand genome RNA is shown there in the middle. They have to come in the cell, they have to be coated with protein in the virion and bring a polymerase into the cell. And these genomes are copied to make mRNAs, and they're copied to make full-length complements for making genomes. Now, there's some very interesting twists here, so let's take a look at them individually. Here's VSV synthesis. The genome is not mRNA. Of course not. It's negative-stranded. It can't be mRNA. Comes in the cell. These viruses enter by endocytosis, low pH catalyzed membrane fusion. The ribonucleoprotein, along with the polymerase, comes into the cytoplasm. And the first thing that happens is the synthesis of subgenomic mRNAs. You can see them here because they're now engaged with ribosomes. So from that single long negative strand genome, you make a series of subgenomic mRNAs, which are translated to make all the viral proteins that are needed, including the RNA polymerase, which is this big protein here called L. This will then replicate the genome. You have to make full-length plus copies, and you copy them again to make minus strands. So the mRNAs obviously can't be copied to make genomes because they're too short. They're not complete copies. So you have to not only make subgenomic RNAs in order to be translated, you have to make a full-length plus strand in order to replicate the genome. And so that's an interesting problem. How do you switch from making mRNA to full-length plus RNA for genome synthesis? Again, we have our bullet-shaped particle. There's the RNA polymerase in the particle, and the nice helical nucleocapsid. The genome is the negative strand color there. And then upon entering in cells, the polymerase makes 1, 2, 3, 4, 5 subgenomic mRNAs, which each are translated to make the proteins that the virus needs. Mostly structural, and M and G are structural, and L is the RNA polymerase. So how does this happen? Here's a schematic of the details to show you how we can switch from mRNA synthesis to genome replication. So here's our negative strand RNA in the middle. So it's come in the cell from the particle. It's coated with proteins. The little circles are the nucleocapsids coating this RNA. The polymerase is bound at the 3' end. It starts to make mRNAs starting right at the 3' end. The polymerase first makes the N RNA, it stops, it reinitiates, it makes the next one, it stops, it reinitiates, it goes all the way down the genome, starting and stopping and making mRNAs. At some point, it's got to stop making mRNAs and make full-length plus strands. Here's a full-length plus strand shown at the bottom here. And the key is, when enough nucleoprotein is made to start coding the plus strand RNA, it serves as a anti-terminator of mRNA synthesis. So you can see as these mRNAs are made early in infection, there's no NP protein made yet. Of course, you can't have NP protein until you have the RNA made. As soon as NP protein starts to be made, those NP proteins will coat the newly synthesized plus strand, and they act as anti-terminators so that the polymerase no longer pauses and makes individual mRNAs. It goes down and copies the minus strand and makes a full-length plus strand. And from that, of course, it can make minus strands, which are going to be incorporated into the genome. So this is all a matter of control of the reactions by the NP protein, shown here in some detail. Here we have the polymerase at the very 3' end of the negative strand RNA. It makes an mRNA for the first gene, the N gene, which is shown there, N mRNA. It terminates and reinitiates for the next one. And that's how you get individual mRNAs. When the amount of NP protein rises, the NP protein coats the first RNA, and it prevents the polymerase from terminating, so it makes simply a long plus strand RNA. And that's shown here in some more detail. Here we have the sequences between the genes, if you will. So there's our minus strand genome. The polymerase has finished the first N mRNA, and there's a stretch of U's in here. This is part of a termination signal for the polymerase. It says stop, so the polymerase stops, but it sees these U's and it starts slipping. And it starts to churn out A's, and that's actually how you get polyadenylation. You can see there's many more A's than there are U's on the template, but you get slippage and recopying of this sequence, and then eventually it stops and you release the mRNA. So that's the mechanism of termination and polyadenylation. It's non-templated synthesis, really, and that's overcome by the presence of NP protein. You inhibit that, NP protein inhibits that and allows full-length plus strands to be made. And in contrast, influenza virus does this on a segment-by-segment basis, but there are some very interesting differences that I want to point out to you. First of all, again, influenza virus genome is not mRNA, it's segmented, it's in 8 pieces, single-stranded minus RNA. This is unusual. The virus enters by endocytosis and low pH catalyzed membrane fusion, and the RNPs come out of the particle and they go into the nucleus. Remember, I showed you a video of that last time. They go through the nuclear pore into the nucleus. In the nucleus, the minus strands are copied by an associated RNA polymerase into mRNAs, and the mRNAs are exported into the cytoplasm where they can be translated into various proteins, including structural proteins to make new virus particles. At some point, you have to make full-length plus strands, and that is shown here in order to make more minus strands. And those minus strands are eventually going to be incorporated into new virus particles. So you're probably thinking, well, it's probably a similar mechanism as VSV, and you'd be right. The nucleoprotein is involved, but there's some interesting twists. Here's the influenza virus genome, the 8 negative strand segments. Again, when they enter cells, they are transcribed by the virion-associated polymerase. There's one polymerase per RNA segment. It's the little sphere at the end of the helical RNA. They're transcribed into messenger RNAs. And those are translated to form one or a couple of proteins. There can be multiple proteins per segment. But these mRNAs are not complete copies of the negative strand genome RNA. They fall short. Just like VSV mRNAs fall very much short of the VSV genome. These fall slightly short at one end, and that's shown here. Here's a schematic of one negative strand RNA segment. The mRNA that's produced from it is shown at the top. It's capped, it's polyadenylated, but it misses about 20 bases from the 3' end of the genome. So that in itself is not very striking, except that you could never use these mRNAs to make new virus particles because they lack the 3' end. Again, you have to figure out a way, instead of making mRNAs, to make a full-length plus strand copy of the minus strand, which you could then use to make more minus strand copies. And that's in fact what the virus polymerase does. And again, it's very similar for VSV. As soon as levels of the nucleoprotein rises, the protein that binds the RNA, it begins to coat the nascent transcript. Then it prevents termination short of the 3' end, and you get a complete plus strand copy. Similar mechanism for both VSV and flu. Now the other twist here, which is quite interesting, is that this is a primer-dependent RNA polymerase. And the primer happens to be a piece of cellular mRNA. It could be any cellular mRNA that's stolen from host cells. And it's basically a small fragment. It's the cap plus 10 to 13 bases of RNA. And so what this virus does, it has an endonuclease in the particle associated with the RNA polymerase, whose sole function is to chop up the 5' ends off of cellular mRNAs and use them as primers. If that isn't Baroque, what is? And that's kind of shown here, host-capped primer. So all the mRNAs of influenza virus, the first 13 bases are derived from you, but they're non-coding, so they don't really do anything except serve as a primer. And that's detailed here. Here's our negative-strand RNA, just the end. The viral polymerase has an endonuclease associated with it that cleaves cellular mRNA. So the brown box at the top is a cell mRNA derived from your host cell. The endonuclease cleaves it around 13 bases from the 5' end. So now we have a capped primer. And a cap, you can see here, is a G attached covalently to the next base by a reverse bond, a 5 to 5' instead of a 5 to 3'. And this is now going to be the primer for synthesis of mRNA. So again, here's our negative strand RNA. Now the primer is going to hybridize to it, and it's going to serve as a primer. And now the polymerase will start copying the negative strand. So every mRNA has this primer incorporated into it. And for that reason, the cell needs to be actively making mRNAs in order for influenza virus to replicate in it. The last part I want to tell you about, which is pretty interesting, is how polyadenylation occurs. Now remember, for VSV, the way polyadenylation occurs is the polymerase reaches that intergenic sequence. It stops because there's a termination signal there. There's a stretch of U's, and the polymerase gets confused and starts to stutter and keeps copying those U's and makes a stretch of poly A. Similar idea here, but there's a different mechanism. Now here's the polymerase made up of 3 different proteins. It is copying a negative strand, which is shown in the olive green. The 5' end of the negative strand is attached to one of the proteins. That's what this red area is. And the active site is shown at the lower red one, and we're making a mRNA. And so what you have to think about here is, instead of this polymerase moving down the template's being pulled through the polymerase. So there's a stationary polymerase. Grab the 3' end of that negative strand and pull it through the active site. And as it passes the active site, you're going to be synthesizing a plus-strand mRNA. And it's capped, of course, because it stole a cap from the host. If this 5' end of the template is attached, then at some point you can't pull the string through anymore, right? So you get to the point where now you're constrained, you can no longer pull through. And I say you can't pull through, but it's not anyone pulling through, obviously. There are forces at work here that we don't quite understand. And then what happens is, when this negative strand can't be pulled through anymore, there's a U stretch in the active site. And the polymerase begins to copy that. And because the template can't move any longer, it makes a stretch of A, and that becomes the poly A. So polyadenylation occurs as this U stretch is reached, and you cannot move the template through the enzyme complex anymore. So 2 really interesting parts of synthesis. The cap is stolen from the host. 3' poly A is made by this unusual mechanism. Our last question is, how are influenza virus and VSV RNA synthesis similar? A, the switch from mRNA to genome RNA is controlled by an RNA binding protein. B, polyadenylation occurs at a short stretch of U residues. C, viral mRNAs are shorter than minus genome RNAs, or all of the above. What we did, most of you got D, all of the above, which is the right answer. They're all right. But some of you just picked one of the 3. But again, the binding protein controls the switch between mRNA to full-length plus. Polyadenylation occurs at a stretch of U, and in both cases, the mRNAs are shorter than the minus genome RNA. So they're all correct answers. I want to tell you about one last kind of RNA virus replication, and that's for the double-stranded RNA viruses, the Reoviruses. And again, they have segmented double-stranded RNA, which has a minus and a plus strand. The plus strand is very much like a messenger RNA. It even has a cap. But again, in this cell, this RNA cannot be translated. So it has to be copied in the cell by a viral polymerase. And the viral polymerase can make messenger RNAs from this double-stranded RNA, which are used to make proteins, or some of those same messenger RNAs, which are complete copies of the minus strand, by the way. Those can be used to make more double-stranded RNA. So let's take a look at how this works. Again, Reovirus, the plus-strand RNA is not accessible by ribosomes. So even though these double-stranded RNAs, and there are many of them in the particle, even though they're naked, the particle still contains an RNA polymerase. And as I said, the polymerase is at each of the turrets, each five-fold axis of symmetry. Remember, these viruses are taken in by endocytosis. They're unusual in that they do not escape the endosome before it fuses with a lysosome. The normal endocytic pathway is materials are brought in from the surface, and then lysosomes fuse, which digest the materials. This virus remains to be digested by the lysosome, and that's because the outer shell is going to be removed. So you can see on that initial infecting particle, there's a blue outer shell and little red bits on top of that as well. That's the outer shell. And then there's an orange-colored inner shell, and then our RNAs. As this is brought into the cell, the outer shell is digested. And you can see eventually you become a core particle, which is just the inner shell, the inner icosahedral shell, and that's able to poke its way out of the endosome. Now, the core particle has double-stranded RNAs, and it has the RNA polymerase. And those RNAs never leave the particle. You can see they're still in there, and they are transcribed in the particle by polymerases that are at each five-fold axis of symmetry. And not only that, but the encoding process has opened up portals at each five-fold axis, so that the mRNAs that are made by these particles can come out of the portals. And that's what you see coming out into the cytoplasm. These little squiggly guys are capped mRNAs that are going to be translated. So those are translated into proteins. New cores are assembled in the cytoplasm. They're loaded with mRNAs, as you can see here. And they're also loaded with RNA polymerase. So the polymerase makes those mRNAs double-stranded in the particle. And then it matures to a full particle and leaves the cell. So interesting here, not only that the outer shell is stripped off by lysosomal enzymes to allow penetration, but the RNA never leaves the particle. The only nucleic acid of these viruses that ends up in the cytosol is the mRNA. And the original double-stranded RNAs remain in the core, and new ones are assembled in a new core. So it's definitely a different way of making genomes and mRNAs. Here's a schematic of the particle. It's a little bigger so you can get an idea what's happening. Here's the native particle that infects the cell. The inner shell, the orange circles. The outer shell are these blue spikes with the red tips. They proceed as the endocytosis occurs through an infectious subviral particle where just the red proteins have been removed. And finally, enzymes remove the outer blue core to form this core particle, which can poke out of the endosome into the cytosol. And at each five-fold axis, again, there's an RNA polymerase that will copy these double-stranded RNAs and make mRNAs that come out from the portal. And each double-stranded RNA segment gives rise to an mRNA, which gives in turn rise to proteins that are needed to build new virus particles and, of course, also encode an RNA polymerase. How do we know this? A number of years ago, someone took reoviruses, added nucleoside triphosphates to cores. So you make cores, you can add triphosphates, they will get into the interior of the particle, and the polymerase will start making mRNAs. And they did a Cryo-EM construction of particles in the act of making mRNA, and that's what's shown here on the left. So you can see the blue is the inner shell, the green is the RNA inside, and then you can see these little pink things poking out at each five-fold axis, right? 1, 2, 3, 4, 5 subunits around the five-fold axis, and that's expanded on the right, and this is what is believed to be RNA coming out of each five-fold axis. That's why they call this a turret. It sticks up let's go back, sticks up at each 5-fold axis, and the RNA is made just below the 5-fold axis. The polymerase sits there, the RNA moves through it, and the mRNA is made and comes out from the 5-fold axis. So it's interesting, there's one RNA polymerase at each 5-fold axis of symmetry. How many 5-fold axes of symmetry are there in these icosahedral particles? There are 12. And guess what? There are never more than 12 double-stranded RNA segments in these viruses. So probably there's one strand per turret or per 5-fold axis because there's a polymerase sitting there. It's a really cool way of making mRNA. So what have we talked about today? These are the topics we've covered about RNA synthesis. We've talked about examples with plus, minus, and double-stranded RNA, polymerase basics, structure, homologies, motifs, where RNA synthesis occurs. You know, for most of the viruses we've talked about, it's in the cytoplasm. The one exception is influenza virus. That has to be in the nucleus. And probably that has to do with the requirement for capped primers derived from host mRNAs. We've talked about these two processes of genome replication and mRNA synthesis. I want you to conceptualize this. You have to make genomes and you have to make mRNAs. For some viruses, it's the same, like polio and flavies. The mRNA is the genome, so you're always doing both. But for other viruses, they're separate processes. VSV, influenza virus, because the mRNAs fall short of the entire genome. So you have to, on the one hand, make mRNA, and then you have to figure out a way to make full-length plus strands in order to replicate. And I talked about mechanism for that as well. And finally, we talked about how flu and VSV add poly-A to their mRNAs by this unique non-templated stuttering reaction. When we return, we're going to talk about transcription first. Now you may say, why don't you talk about DNA synthesis of DNA viruses? Because when a DNA genome comes in the cell, that's not the first thing that happens. The first thing that has to happen is synthesis of at least one viral protein. Otherwise, DNA synthesis will not occur. So we're going to take up next week DNA-directed RNA synthesis.